So before we get into the nitty-gritty of the steps in those enzyme-controlled reactions of respiration, I thought it was probably worth doing a bit of an overview. Of respiration, so that you know where the stuff goes on. So remember, in the last video I said, okay, so we know from the core concepts part and doing cells that aerobic respiration takes place in the mitochondrion, where ATP is made. So this is my mitochondrion, just to remind you of its structure. We've got that inner membrane folded into cristae, big surface area. We've got the outer smooth membrane. And in between we've got the intermembrane space. I'm going to kind of ignore the DNA and the RNA and all the rest of it, and just label the bits that are going to be relevant to you. And of course we've got that fluid-filled, enzyme-rich, it could have something to do with the respiration area called the matrix. And remember that outside we've got the cytoplasm, and outside of that, sort of separating it out from the cell contents, we've got our cell membrane. And this is going to be the place where our glucose is entering. And of course our oxygen is entering. So remember, oxygen is going to get in by straightforward diffusion. And we know that the oxygen, because mitochondria do the aerobic bit, is going to be diffusing straight into the mitochondria if it's present. Glucose, of course, it will be coming into the cells, usually co-transport. With sodium ions, and that's coming in by facilitated diffusion. Okay, so what goes on in the cytoplasm? So in the cytoplasm, all the cytoplasm reaction is about is splitting the glucose up. And so in the cytoplasm we do a series of reactions called glucose splitting, glycolysis. And to do that, we don't need any oxygen. To split up glucose, don't need any oxygen. The only place where we need oxygen is in here. So, we're starting off with glucose. We're making it into 2,3-carbon sugars, and then we're converting each of those into pyruvate. And again, I'll do those reactions in more detail. So remembering that the whole point of respiration is to make some ATP, here we get a net gain, overall gain of 2 ATP per molecule of glucose. And we're making 2 3-carbon molecules of pyruvate. So pyruvate has 3 carbons. Again, most frequently asked question about the booklet was, what does this 3C mean? And we also make some of this reduced, because we're oxidizing our substrate, we're also making some reduced NAD. So, our pyruvate then diffuses through to the matrix if oxygen is present, where it gets converted into a 2-carbon molecule. I like to call it acetyl, but you can call it acetate, it doesn't really matter. And acetyl is then helped into Krebs cycle and again there'll be a more detailed video. And throughout these throughout this sort of series of reactions, we're going to release some carbon dioxide, which will diffuse out, and then of course will diffuse out of the cell entirely. And we're going to make an ATP. That will certainly be handy, that's the whole, yeah, let's do that. And we're also going to do a lot of those dehydrogenation reactions, so we are also going to be removing hydrogen and passing it to our coenzyme. And we're going to use a different coenzyme to carry one of those hydrogens, and that different coenzyme is FAD. So we're going to be taking the hydrogen away on those 3. So that's kind of the overview. And remember that I said that their job, the job of these coenzymes was to carry hydrogen to the electron transfer chain. So we also have, I'm running out of colours, I don't know, pale blue will do. We also have electron transfer chains, and they take place on the cristae. So we've got carriers and proton pumps. What their job is that hydrogen, remember this is an overview, I will do it in more detail in different videos. Our hydrogen is going to split off into a proton and a high energy electron. The electron is going to go down the chain, releasing energy to pump those protons across into the intermembrane space. And then of course we're going to make ATP with that, so in addition we've got stalked particles through which those protons can diffuse and release ATP. So you're thinking back now to the last video where I said, and the overall equation, we can see that the carbon dioxide is coming off, but where's all the water coming from? It's coming from the end of the electron transport chain. So at the end of the electron transport chain, when we've transported the hydrogen electrons through, we're going to add oxygen to make water, which again, if there's an osmotic gradient, can leave the mitochondrion. So that's the kind of overview. So you need to know that the cytoplasm does the reactions of glycolysis, no oxygen required, that everything that goes on in the mitochondria requires oxygen, but it's only actually used at the end of the electron transfer chain. And the idea here is if you haven't got oxygen, you can't accept the electrons and protons at the end, and therefore that sort of backs up, which means no more hydrogen can be removed around here, so these reactions back up, until you're not getting rid of the pyruvate, and you won't have a diffusion gradient across, so you can't then diffuse it into the mitochondrion. So, names of reactions: pyruvate to acetate, link because it joins glycolysis to Krebs, and the circular one, Krebs cycle. And then over here on the Christie, we've got the electron transport chain. And those in blue. So Christie is the site of the electron transport chain. And of course we've got our stalked particles, which are the site of chemiosmosis, the manufacture of ATP using that proton gradient. So I think now we need to really get into the detail of what's going on in each one of those stages.